Welcome back, everyone. So, today we're diving into a foundational explainer on cell biology. We're literally unpacking the absolute building blocks of everything that breathes, moves, and grows. Whether you're a seasoned science enthusiast or you're just genuinely curious about how life operates at that microscopic level, well, you're absolutely in the right place. Let's get into it. To kick things off, I want you to consider this high stakes truth. You might forget your name, your address, or your PIN code, but you can never forget that the cell is the structural and functional unit of life. It really sets the perfect tone, doesn't it? We're talking about the absolute, non negotiable basic unit of our existence. Because frankly, you just can't understand a complex organism without first understanding the engine that actually drives it. Okay, let's dive into this. Here is our roadmap for the explainer. 1. Introduction to the cell. 2. The cell theory. 3. Sizing up the cells. 4. Prokaryotes, the simple cells. 5. Eukaryotes and cell membranes. And finally, number 6, the endomembrane system. Section 1. Introduction to the cell, the structural and functional foundation. So, the absolutely crucial point here is this definition. A cell is the basic, independent entity that performs all essential life functions. It is the defining feature that separates living organisms from non-living objects. I mean, if an entity doesn't have a cell, it just cannot be considered alive in the traditional sense. And actually, this perfectly illustrates why viruses aren't strictly classified as living organisms. They literally lack their own independent cellular machinery to survive and reproduce. Wild, right? Moving right along to Section 2, the cell theory, a historical journey of discovery. So let's walk through this timeline because it's actually super fascinating. Back in 1665, Robert Hooke first discovered the dead cell by looking at a piece of corkwood. Then, decades later, in 1831, Robert Brown discovered the nucleus, which is essentially the brain of the cell. But things really heated up in 1838 and 1839 when a botanist, Schleiden, and a zoologist, Schwann, teamed up to propose the original cell theory. They figured out all plants and animals are made of cells. But, they actually had one major gap in their theory. They couldn't explain exactly how new cells formed. And that is where Rudolf Virchow steps in during 1855, and he completes the whole theory with this famous Latin phrase, omnis cellula e cellula, simply meaning, all new cells only come from pre-existing ones. All right, Section 3, sizing up the cells, visualizing scale and diversity. Now, what's really interesting here is the sheer physical diversity. We're talking extremes. We go from the incredibly tiny mycoplasma, which measures just zero point 0.3 micrometers in length, all the way up to typical bacteria, and then eukaryotes. But pay attention to those bottom two rows. The nerve cell is the longest cell in the human body. It can literally be over a meter long, acting like this giant thin wire transmitting signals. But then, look at the ostrich egg. That is the largest overall single cell, meaning it has the absolute most massive volume. Length versus volume. It's a really crucial distinction to keep in mind when we're talking about these cellular extremes. Now, there are some pretty fundamental differences between plant and animal cells. And there's actually a brilliant memory trick for this: the 4 C's. Plant cells possess 3 unique C's: a cell wall for rigidity, a central vacuole that takes up a ton of space, and chloroplasts for photosynthesis. But what about animal cells? Well, they possess a unique 4th C, centrioles or centrosomes. These are completely absent in plants and are primarily used to help the animal cell divide. Lock those 4 C's into your memory and honestly, you'll never confuse the two again. This brings us to Section 4, prokaryotes, the simple cells and masters of microscopic efficiency. Despite lacking a well-defined nucleus, prokaryotes are absolutely the masters of microscopic efficiency. Let's break down their armor, the bacterial cell envelope. It has 3 distinct layers. So the outermost is the glycocalyx. It's a carbohydrate coat that acts as a first line of defense, and it can be a loose slime layer or a really tough capsule. Next, we have the cell wall. It's perfectly compared to something like a thick layer of concrete. It's this highly rigid, tough middle layer providing the structural support to keep the cell from literally bursting open. And finally, the inner cell membrane. This is the intelligent gatekeeper that decides exactly what enters and exits the cell. So it begs a question: what happens if a cell completely lacks that rigid cement wall? Well, you get this fascinating organism, mycoplasma. It's playfully known as the joker of the plant kingdom. Because it completely lacks that rigid cell wall, it's described as pleomorphic. Kind of like how a Joker changes costumes to entertain, mycoplasma can continuously change its physical shape to adapt. It is literally the ultimate microscopic shapeshifter. While we're on the topic of these cellular components, let's look at the protein factories, the ribosomes. You're going to notice some completely counterintuitive math here. Prokaryotic ribosomes are 70S, and they're made of 50S and 30S subunits. Wait a second, 50 plus 30 equals 80, right? And for eukaryotes, the 80S ribosome is made of 60S and 40S subunits. Okay, so the trick here is that the S stands for Svedberg unit. It measures density and how fast things settle in a centrifuge, not the actual physical weight. Think of it this way. If you wrap a very light, fluffy blanket around your solid body, your combined density isn't just a direct addition of both weights, it averages out. And that is exactly why 50 plus 30 yields a combined density of 70S. Makes sense, right? Let's move to and see how this builds in Section 5, eukaryotes and cell membranes, compartments and gateways. Everything about how a complex cell protects itself really revolves around this pivotal concept, the fluid mosaic model. Proposed by Singer and Nicholson back in 1972, it describes the cell membrane so perfectly. The fluid part refers to this flexible, flowing lipid bilayer. And this fluid nature is absolutely essential. It's what allows the cell membrane to stretch, to grow, and even to pinch off during cell division. Then the mosaic part refers to the various proteins scattered throughout this lipid sea. It's kind of like raisins floating in a bowl of pudding. These proteins are incredibly important because they act as vital transport channels, essentially shuttling all the necessary molecules in and out of the cell. All right. Almost at the end. Section 6, the endomembrane system, the cellular factory floor. Let's explore how these highly compartmentalized eukaryotic organelles actually work together. It is a perfectly choreographed sequence. I want you to think of this system as an incredibly efficient manufacturing plant. So step 1 is the endoplasmic reticulum, or the ER. This is where the raw materials, your proteins and lipids, are physically produced. Step 2, these raw materials are shipped right over to the Golgi complex. This is the packaging department. Materials are sorted, they're modified, and prepped for their final destination. And then finally, step 3 involves the lysosomes and vacuoles. These act as the recycling centers for digestion and the warehouses for cellular storage. It's just this continuous, beautifully orchestrated biological workflow. I actually want to zero in on the Golgi complex for just a moment. It's the cell's main shipping and tagging center. It performs a highly specific biological process called glycosylation. Now, in simple terms, this just means adding specific carbohydrate tags to the newly made proteins and lipids. It is literally the microscopic equivalent of stamping an address label onto a package. Without these specific sugar tags, the cell's transport network wouldn't have a clue whether to ship a protein out of the cell, embed it into the membrane, or drop it into a lysosome. It's a real game changer for cellular logistics. Which leaves us with this final, really provocative thought. If a single bacterial cell, like a simple prokaryote, has absolutely no complex organelles, no advanced endomembrane system, and no cellular factory floor like the one we just saw, well, how does it still manage advanced metabolic functions like cellular respiration and even develop complex antibiotic resistance all on its own? I mean, it really prompts us to marvel at just how much brilliant survival machinery is packed into a single, seemingly simple drop of life. There is still so much more to uncover about these microscopic marvels. But for now, keep questioning, keep learning, and I'll see you on the next explainer.